Since I want to make biology and chemistry videos, I figured I would try to discuss pH a little bit between those two disciplines. I know pH can seem like an intimidating concept, but hopefully this will clear it up at least a little bit for you. So, the importance of pH, especially in biochemistry, is that water is the solvent of life. And water has got this interesting equilibrium you can see at the top of the page, where it turns into hydronium ion, or H3O+, and hydroxide ion, OH-, by the exchange of a proton, or an H+. Sometimes you will see this equation written like this. Oops, I forgot my equilibrium. But this H+ and this H3O+, they're basically synonymous. The difference is that H+ is what gets exchanged between things, while H3O+ is the species that actually exists in aqueous environments. So, if you've taken chemistry, or at least part of chemistry, you might know that the equilibrium constant of a reaction is the products to the power of their coefficients over the reactants to the power of their coefficients. But the important thing is that liquids and solids are not included in equilibrium constants because they're considered to be in so much excess that it would just, throw things off. So, for the purposes of the equilibrium constant, we are not going to use water. And the equilibrium constant will then be H3O+ to the first power times OH- to the first power, and to the first power can just be invisible because it cancels out. And the equilibrium constant for water has been determined experimentally to be 10 to the negative 14. So where does this come in pH? Well, P means negative log in science. So pH is the negative log of the concentration of H3O+, or negative log of the concentration of H+. Again, those two things are synonymous. So when water has got nothing else in it, if you start with only water and you progress this reaction until it gets to equilibrium, then what you're going to end up with is logically an equal mix of hydronium and hydroxide ions. So if hydronium times hydroxide equals 10 to the negative 14 and hydronium equals hydroxide concentration, then that means the hydronium, or H+ concentration, is going to be 10 to the negative 7th power. So if we take the negative log of 10 to the negative 7th power, we get 7, and that is our neutral pH, is pH 7. Now there have been a couple different theories on what exactly acids and bases are in terms of pH. And they're named after a couple of dudes who did experiments on acids and bases. So the earliest one was Arrhenius, and he thought that acids were proton donors and bases were hydroxide donors. Now a proton is just an H+, because remember, hydrogen is the first element in the periodic table. It's got one proton, one electron. So if you have a B+ and take away the one electron, it is simply a proton. Anyways, then Bronsted and Lowry came along and they said, well, actually, an acid is an H+ donor and a base is not an OH- donor always. It is more an H+ acceptor. And then Lewis came along and modified the definition even more by saying that an acid is an electron acceptor, whereas a base is an electron donor. An electron is signified by E-. So the way that these definitions kind of work is that the biggest umbrella is the Lewis acid and base definition. Within the Lewis acid and base definition is the Bronsted-Lowry acid and base definition. And then within that is the Arrhenius definition. So all Arrhenius acids and bases are also Bronsted-Lowry acids and bases, and all Bronsted-Lowry acids and bases are also Lewis acids and bases. What you're going to see as you do more biology and chemistry is that in organic chemistry, we like to use the Lewis definition of acids and bases, whereas in general chemistry and in biochemistry, we like to use the Bronsted-Lowry definition. So the Bronsted-Lowry definition is what we're going with today. So based on the definition that pH is negative log of H+, This is the graph for y equals negative log of x, and we can see that as x goes up, the negative log of x goes down. So the pH scale is kind of backwards. When you've got a high H plus concentration, as you will when you have an acid, because remember an acid is an H plus donor, you are going to have low pH. If you have a base, which is an H plus acceptor, that's going to have lower H plus because the base is soaking it all up, and you're going to have high pH. So ignore the left side of the screen for now and just look at this pH scale here. So you can see that on the acidic end, we have things like citrus fruits, like carbonated beverages, and that one we're going to explain in a second. Whereas physiological pH, like blood and stuff, is generally a little above neutral. I mean, you have parts of your body, like your stomach, that are going to be acidic, but most of you is slightly basic. And then at the very basic end, we have things like cleaners, like bleaches and stuff. Now, another important concept in pH is buffers. So a buffer is going to be something that helps maintain the pH. And in order to understand the concept of a buffer, we have to look a little bit at acid-base reactions and how an acid and a base work with water. So usually when people teach acids and bases, we use a hypothetical equation in which an acid is represented as HA. In this case, the A is the rest of the molecule, and the H is going to be the proton that comes off. So we combine this with water, and it's in equilibrium with H3O plus, which is what you get when you have an acid, and A minus, because we took off an H plus, and so for conservation of charge, we need to have it be A minus. So this A minus is called the conjugate base of the acid HA. That just means that they're only different by one proton. And vice versa, if we're going to write it like A minus plus H2O is in equilibrium with HA and OH minus, then HA is the conjugate acid of A minus. So whenever you have an acid, it's in equilibrium with its conjugate base, and whenever you have a base, it's in equilibrium with its conjugate acid. For strong acids, what happens is the equilibrium is really driven to the right, to the products, and you don't get a lot of stuff coming back the other way. For strong bases, it's going to be the same thing, that the equilibrium is going to be very product favored. But for weak stuff, it's very easy to put something in and via Le Chatelier's principle, change up the equilibrium. Remember, Le Chatelier's principle is like if you do one thing, you want to balance it out. So for example, if you look at the top equation, if I put in more HA, this equation is going to go toward the products to soak up the HA and to have a more equal balance of the reactants and products again. So that's the concept of a buffer, is basically that you have a weak acid and its conjugate base, and if you put in acid, it gets soaked up by the base, and if you put in base, it gets soaked up by the acid. And when I say soaked up, I mean reacts with it and neutralizes it. So what is the biological significance of buffers and pH maintenance? Well, there's a famous buffer in your blood called the bicarbonate buffer, and it has to do with how does your body know when and how fast you have to breathe. Well, you breathe in oxygen, you need that for your cellular respiration to make energy for your cells, and you breathe out carbon dioxide, which is a byproduct of cellular respiration, right? So when you have carbon dioxide from doing a lot of exertion and doing a lot of cellular respiration, if you have a lot of carbon dioxide in your blood, it's going to react with water because there's water all around in your body, and it's going to form H2CO3 or carbonic acid. So as your blood gets more CO2 in it, it gets more acidic. And this carbonic acid is going to be in equilibrium with a conjugate base of HCO3 minus or bicarbonate. So if your blood gets more acidic, it drives the equilibrium toward bicarbonate, and if your blood happens to get more basic from a lack of CO2, the equilibrium gets driven back the other way. And blood pH is how you tell when to breathe, because your blood interfaces with your cerebrospinal fluid, and that goes into your brainstem, and your brainstem has sensors that can tell when things are getting a little too acidic, when the buffer isn't doing enough, and you have too much CO2 and therefore too much carbonic acid in your blood, and it says, hey, lungs, you gotta breathe. So that's buffers. And there's one more topic I want to look at in the vein of pH in biochemistry, and that is amino acids. So, as we know, if we've watched the protein video, amino acids kind of have 4 components. They have an alpha carbon, a hydrogen, which I don't count as one of the 4 components because it's just a hydrogen, an amino group, and a carboxylic acid group, and a side chain. So, the interesting thing about amino acids is even though we can write this all neutral like that, what really happens to it is it ionizes. So the NH2 group is basic, and it's going to end up accepting an H+. So an H and a plus. The carboxylic acid group is acidic, like it says in the name. So it's going to lose a proton, so it's going to lose an H and lose a plus, aka gain a negative charge. So that's the significance of amino and acid, is that they actually, in biology, in biological environments, bases gain positive charge and acids gain negative charge. And this amino acid that is ionized in plus and in minus is called a zwitterion. Ion meaning ion, and zwitter being German for 2. Now beyond that, the R group or side chain can actually also gain a charge from being acidic or basic. There's 2 acidic amino acids. There's aspartic acid and glutamic acid. And so we can see that here where there used to be a hydrogen, now we have a negative sign because the H plus went away. And we still had the zwitterions here, like we talked about. There's 3 basic amino acids. So there's lysine, there's arginine. I might have mixed those 2 up. I'm not sure. It's been a while, guys. Okay, and then there is histidine. And these guys all have basic amino groups in the side chain, and they get positively charged as a result. So when you have a fully fledged protein and you're looking at tertiary structure, and you know how we talked about in tertiary structure there can be ionic reactions? This is why, because you have basic and acidic amino acids. And then beyond tertiary structure, these amino acids can also contribute to chemical reactions if they're in the active site of an enzyme. They can attract things that are the opposite charge from them and keep the substrate in place and help to facilitate the reaction that turns it into a product. So that's the basics of pH. I want to get to more videos on titration. I want to get to more videos on electrophoresing proteins to tell the charges on the amino acids based on pH. But that's for another day. I hope this helped at least somewhat to understand pH.